Oh, sorry. Good morning or afternoon. I'm Danielle McDermott, and I'll go ahead and get started and see if we have more participants as the time progresses. So I'm a neurologist, an epileptologist. I'm also a director in clinical development, and I'll use this lens to discuss the drug development and clinical trials from a kind of a high level, and specifically how patients can better understand the clinical trials process and understand how patients can proceed through the process. In terms of a conflict of interest, I'm currently employed by Radius Health. I also have consulting agreements previously with NeuroPace and SK Life Sciences. So just to back up and think about why clinical trials are important. And so also please feel free to, as we're going along, put any questions in the chat as well, since we're a pretty small group here. So you know, I want to just talk about why trials are important, and you know, that we know that although you know, trials don't always lead to implementation of a new medication or a new intervention, they may provide benefit to the patient from a new treatment that was not available previously. Also importantly, trials may not provide the opportunity for the person themselves, but they provide benefit and potential access of treatment to others. And as I said, they may not represent improvement of the standard treatment, but knowledge of a disease, a condition, or understanding how, just as important as response to treatment, non-response to treatment is also valuable in terms of knowledge gained through clinical trials. So in terms of, you know, putting this together, we have basic science research that helps us formulate questions that then leads to preclinical research. And what do I mean by preclinical? That's done in a laboratory setting, typically with animal research before we test any intervention in humans, to evaluate first safety and tolerability. And then on a larger scale, look at you know, the intervention in larger groups of people that are typically have a certain defined disease state. And then after a medication or a treatment may be approved, there's called a phase 4, where we look at optimizing that therapy. And this continues to happen on a cycle, because after a treatment may be approved, it may lead to further research questions or ways to optimize, you know, current existing therapies that then will expand on basic research questions, and the cycle then will repeat itself. So what is a clinical study? So big picture, clinical studies basically involve the research of human volunteers. And in order to gain medical knowledge, we look at what are the unmet needs, what are knowledge gaps, and we look at a goal of adding to that existing medical knowledge in order to gain improvement in treatment. Clinical studies are the core of medical research. There's two types. There's observational studies, and then there's interventional studies. And interventional studies are what we also refer to as clinical trials. So looking at this in a longitudinal fashion, when we look at from the laboratory to approval. So this can take over a decade from the initial discovery of a molecule, for example, in the case of a drug, until the potential final approval of a drug. And the drug must pass several phases prior to clinical trial, even the application to study that drug by regulatory authorities. In order to apply for a clinical trial, the investigator, or sorry, the sponsor undergoes what's called an IND in the United States. It's called an application for an investigational new drug. In Europe, it's a clinical trial application. And so that process involves information such as all of the preclinical information, so anything that was done in the laboratory, all the manufacturing data, and any clinical information that is known from similar medications or treatments. And at the same time, a protocol, a study design, is reviewed by an independent ethics committee. And in the United States, it's referred to as the Institutional Review Board, or IRB, that looks at the study protocol and says, is this done ethically? Does this make sense? Are there any concerns in terms of the risk versus the benefit, and so forth? When we look at an observational study, it's exactly what it sounds like. It observes people in a usual setting. So the researcher will gather information, groups of study participants according to their broad characteristics, or so-called demographics, and then we'll compare changes over time. So there's no intervention, it's purely observation. So again, it looks at health outcomes. And again, there's still a study protocol. It's how the data is going to be collected. It's what the investigator is going to look at in this particular group of patients that share some similarities. But they are not assigned an intervention, such as a medication or a dietary treatment, for example. But there is value in observational studies because certain patients with certain common characteristics or may have similar traits that can then be evaluated in an investigational study. For example, an investigator may observe the effect of different lifestyles on bone health in older adults. So may make associations with, say, exercise and bone health, or being sedentary and bone health, diet and bone health. So these are things that are non-interventional, but may have an effect. In terms of a clinical trial, this is differentiated by the intervention. So this can be a drug, it can be a device, a surgical procedure, it could be a diet, and there is an endpoint or an outcome that is measured. The evaluation is either safety or the effectiveness, or both. And then there's a comparison that is made. The comparison is either what's available, and that's considered standard therapy. Placebo, which is something that looks like the interventional compound, but actually has no active medication in it, or no intervention at all. So the patient may know that they're not having an intervention. So maybe, for example, to have a diet versus no diet. It must be designed, however, carefully to provide the most information and so that there is the least possible risk to the participant. Trials are described by the different phases, and I will go into those in depth. When we look into the investigation, we want to know what we want to investigate the outcome of the intervention. So we have to control for potential bias and variation within the patient population so that we know, when we're talking about epilepsy, for example, that no two people with epilepsy are the same. So how do we account for those differences within our patients, and how do we control for ages, how do we control for differences in environments, and so forth. So we have to be very careful about patient selection. We have to also be careful that we include different kinds of patients. So these are very challenging things when we're coming up with the investigation plan. But we begin by asking a research question and then come up with a testable hypothesis. So how do we come up with an idea of what we think might be the outcome? So there's two ways that we can investigate the question. So one, which is the most common way, is that we test what's called the null hypothesis. So that assumes that there's no difference between the new therapy and the standard. And so we seek to disprove the effect. So we construct the alternative hypothesis, and I'll come up with some examples. The opposite is an estimation. So you estimate what might be the response rate between two therapies. So one may be the new therapy versus the standard therapy. So we then have to define and clarify this research question. So it's important in order to produce high quality evidence. So with regard to the null hypothesis, we can't just say, does this medication work? That's not a scientific way of evaluating a research question. What is a different way of going about this is saying, the null hypothesis would say medication X is no more effective than the standard of care. So we're saying there's no difference between these medications. In an estimation method, we would say what proportion of patients taking medication X attain more than 50% of seizure reduction compared to patients taking medication Y. So we're directly comparing two medications with an outcome. So we go from refining the research question, and then we have a large project team. Once the research question is understood, we want to then further refine what we are evaluating in terms of the condition. So what characteristics of the condition are we evaluating? What characteristics of the therapy are we evaluating? What are the available therapies? What are the measurement instruments, and what are the outcomes and the endpoints? So for example, when we talk about the world of epilepsy, we would need to refine what kind of epilepsy we're evaluating. So we would refine the type of seizures that we may want to evaluate, or more broadly, we may look at conditions that may be associated with seizures. We would look at the type of therapy that we're using. So what kind of therapy or what type of characteristics does the medication or the device have that is different from other therapies that are available? What patients are being quote, targeted, meaning what is the need and is there a gap in the availability of certain therapies to patients? How are we going to measure outcomes? So are we going to purely count number of seizures? Are we going to measure quality of life? How are we going to do this? There are many different types of scales or endpoints that we can measure in clinical trials that are validated, meaning that they've kind of had to pass a litmus test and to be shown that they can be replicated in the patients that we're treating, and in order to have scientific rigor. So the trials are classified into phases based on the objectives of the trials. And this may seem pretty simplistic in terms of the nomenclature, but we just basically go from phase 1 to 2 to 3 to 4. And I will go through these one at a time. Before we get to the phase 1, I alluded to the fact that there's preclinical data that informs the phase 1 trial. So the preclinical trial or phase 0 trial is typically conducted in animals to determine if a new medication or intervention, a drug, a procedure, or surgical intervention is likely to be beneficial and is likely to be safe in order to proceed for testing in humans. So we need to have that information in order to proceed with the next phase 1, which is the first studies of an intervention that is conducted in humans. It's important to know that the phase 1 trial are often very small sample sizes, meaning typically 20 to 80 patients. And often phase 1 trials are not conducted with the patient population that is attempted, that is the quote target population, as I mentioned. So if it's an epilepsy trial, for example, we may not evaluate this in patients who have epilepsy. It may be conducted with patients who are who don't have epilepsy and typically may not have any chronic condition whatsoever. We want to evaluate the safety, any drug interaction, and the bioactivity. So how is the medication metabolized by the body? And about 70% move to the next phase. In terms of a phase 2 trial, if the intervention is successful in moving on to the next phase, we then use preliminary data on whether an intervention works on people with certain condition or disease, and compare it again, I said, to placebo, meaning an inactive compound or a different drug, which is thought to be a standard of care. We also want to see what perhaps is the optimal dosing. So we may test 1, 2, or 3 doses to see if there's an optimized dose where patients may respond preferentially. So if there's no improved response at a high dose compared to a low dose, there may be no advantage in exposing patients to a high dose when the same outcome can be achieved with a lower dose. And we're always investigating safety issues all along in every phase, but this is the pinnacle of the phase 2 trial, especially in patients who have the condition that's has the intended outcome. In terms of phase 3 trials, phase 3 trials are generally large. Many study participants And again, as I said, we're looking for diverse populations to enrich the study. So meaning that we want equal representation of male and females, of different ethnicities, to recruit from, not just the United States, but around the world. And it's designed in order to, confirm is used generously here, but how effective the intervention is. Again, we're looking to gather more information about safety, more information about dosing, and for potentially combination dosing. And sometimes these are referred to as confirmatory trials or registration trials, especially in the context of pharmaceutical development. So to review, in phase 1, so these are post-clinical, or clinical, I should say, post-preclinical. Phase 1 would be the first studies that are in humans, small sample sizes with non-affected or healthy participants. And we're looking at things like pharmacokinetics, which are how is the drug metabolized, pharmacodynamics, what are there any drug interactions, and we're looking at safety, so any toxicity. Phase 2 is typically looking at dose response, so optimizing dosing, safety issues, and preliminary evaluation of efficacy. So does this work in the intended patient group that we are evaluating? And then phase 3 is a larger trial to further evaluate efficacy and safety in the target population. So a phase 4 trial is conducted after FDA registration or approval of an interventional product. And we are gathering more information about safety and efficacy, or how to optimally use a product. So maybe there's a way of dosing it. Maybe it's once a day, twice a day. Maybe we're comparing that. Maybe we're looking at whether a product should be is better administered with food or without food. So there's things like this that we do after the drug may be approved. This is often recommended. These types of studies may be recommended by the FDA after approval to further refine how physicians and clinicians may better, direct therapy. So we also are evaluating the treatment over a long time. So these are often called open label extension. So for surveillance. So always looking at ways to track patients who had been involved in the initial studies and continuing to evaluate them. In terms of what percentage of trials continue to the next phase. So in terms of the phase 1, I went over this briefly, about 70%. In terms of phase 2, this is the big what we call attrition, or where the dropout is. About 40% of phase 2s continue to the next phase, and then about 80% in phase 3. So it's quite an arduous and long process, as mentioned, with a drop off along the way. So what are the challenges in evaluating neurological outcomes? And I alluded to some of these earlier, but the outcomes, meaning a lot of the things that we're studying or the endpoints can be subject to a lot of variation. So for example, if we're studying not just seizures, which I guess can be actually very difficult at times to study as an endpoint, but we also want to see, not just does a patient have an improvement in seizures, but does it impede, what are other things that a patient may find beneficial in terms of outcome in relation to an intervention with regard to an epilepsy study. So things that I can think of would be quality of life measures. And these can be very subjective. And we may be talking about things like cognition. So how is cognition, either improved, or how is it, affected by a certain intervention? So that can be very subjective. It may be, very different in one patient's experience related to another. Same thing in terms of the lack of a gold standard definition for certain neurological diagnoses. So, for example, neuropathy. Sometimes this is a very broad category, but there are very specific ways to study neuropathy, and there are very kind of broad ways to look at neuropathies. Oops. Sorry about that. The outcomes can be high dimensional, meaning that we can't necessarily capture outcomes using a single numeric score. So, for example, if we want to study a seizure count, that can be something that can be captured by a numeric score, 1, 2, 3, or 4. But say we want to look at MRI information. That's very difficult to quantify, so that's very difficult to have a input into it, again, to translate this. So an example, think of pain in a clinical trial that may be used to evaluate a medication that's being used to treat pain. We may consider the nature of the pain and how different individuals may describe pain. So they may think, well, how is that scale from 1 to 10 of pain different? How may they describe it? And what is the placebo effect when it comes to individuals that may be taking a medication on a clinical trial for pain? Isolate the effect of a single medication when we have all of these factors that are coming to play treating one individual, suffice to say that we have many individuals participating in a trial. So we have measures called good clinical practice to try to preserve the integrity of our scientific quality and design. And this involves lengthy documentation. There are many regulations depending on the part of the world where you live, but this all derives from the following documents that I've listed here, but it is recognized as a responsibility of all of the entities involved. So the sponsors being the company that has kind of conceived of the intervention. So the investigator, which is the physician or physician assistant, or so that is the person that is working with the sponsor at a clinical trial site to employ that clinical trial. And then the ethics committees as well to ensure the safety of the patients. So this has all been a work in progress over I would say decades to ensure that we are doing ethical things to preserve the rights of patients involved. In addition, many experts are involved behind the scenes who must be trained in good practice. So I listed only a fraction of these, but We have, clinical research associates, and clinical operations experts, statisticians, manufacturing experts, regulatory experts, toxicologists, pharmacologists, project managers, clinical scientists, all the way to hospital pharmacies, clinical laboratories, you name it, but all are responsible for ensuring these good practices in all of their respective roles to ensure that the best science with the best integrity is being carried forward. Again, protecting human subjects, it's to be reinforced that all individuals are responsible for protecting subjects who, again, volunteer to participate. Informed consent is one of these procedures where it's essential to inform all prospective subjects. This creates some challenges, more than some, especially when we're referring to children, persons with disabilities, persons who maybe, for example, prisoners who may want to participate in research. And so there's a responsibility to ensure that their data is safeguarded. I have already touched on equitable selection of subjects for research. So are we doing our due diligence to ensure that we're selecting underserved and underdeveloped populations and reaching out to these areas and not just continuing to go to the same kind of demographics or clinical sites even that we have gone to over and over again. What are the rights of research subjects? For example, is a subject, so should they be compensated for participating in a clinical trial, and how so? What if a subject is injured through the process of a trial? What is their recourse for this? These are all very complicated and questions, but I think they're very important to think about, and especially if any of us are involved in any capacity in a clinical trial. So how does a patient arrive at a clinical trial? What is the pathway? What can one expect? And in general, I'll go through each of these, but it begins with a diagnosis of the condition, some sort of a referral, whether or not this is the patient finding about the trial on their own or a healthcare provider informing the patient of the trial, to pre-screening, to the informed consent process, to being screened, then randomized, and then finally trial participation. So there's a process that goes into play long before the patient can actually participate in the clinical trial. So what about the diagnosis and referral? So the diagnosis part should be relatively straightforward. Hopefully the patient understands their diagnosis, and a physician or healthcare provider may refer the patient to the trial or trial center. The healthcare team at the center may discuss the availability of the trials at their center as well. And the patient obtains information by doing research. So for example, I'll show you some different websites, but one example is Clinicaltrials.gov. So patients may be very motivated to participate in a clinical trial and can look and see what's available for their certain diagnosis. So the pre-screening process. So this is an initial review of records for trial criteria. Specifically, what I mean is inclusion and exclusion criteria. So once the patient has identified a trial that they may be interested in, perhaps their physician has said you may be a good candidate for this, they may meet with the trial team and have an initial review, say, well, are you over 18 or are you between 6 and 18? So there may be a certain age criteria. There may be other qualification criteria that are being evaluated in a specific study. And so these are kind of pre-screening to make sure that the patient can go on with evaluation. So in terms of screening, then the patient would undergo testing and some preliminary evaluation to assess further eligibility for this study. So for example, there may be a review of medical records to say, so the patient has a history of a certain type of epilepsy. Let's make sure through verification of a medical record that this is verified. That's just an example. So looking at then randomization. So this is done for control trials where one group of patients is receiving the intervention or active treatment, and the other group of patients is receiving, is in the control arm. So they would receive placebo or standard of care, or placebo may be equivalent to no treatment, but in some cases, we had talked about a little bit about interventional studies that may not be a medication, for example, like exercise or some other sort of an intervention. And then finally, the patient then participates in the trial. And so the patient is followed by the trial team, and there's an interface between research and clinical care. So I think, yeah. So there's a very clear outline of study visits that is provided at the time that the patient is enrolled in the trial. The patient is consented. There are some, at that time, the patient would understand very clearly what the potential risks are and what the study outline is. So for example, some studies are very intensive and may have weekly, for example, visits, whereas some are less intensive and may have visits every 4 weeks. So it really is important that patients understand what the study visits look like, what the outline of the study is prior to participating. Some studies may, especially when we talk about epilepsy type of studies, may require lots of diaries to document, seizures, or may require being able to maintain or fill out questionnaires. So it's important that the person participating is able and willing to participate in this. So what are some ways to see what resources are there? One is the National Institutes of Health. And so this can give patients information about participating in clinical trials. Why should you participate in a clinical trial? This is a simple link to this website is nih.gov. And more specifically, finding clinical trials near you, I will give you some epilepsy links. So I really liked this slide because it's kind of catchy, and I thought, well, so clinicaltrials.gov is one. What is clinicaltrials.gov? And I'll show you a screenshot of this. This is a searchable registry. And the results of federally and privately supported trials conducted in the US and around the world. Provides information about what is the purpose of the trial, who might participate, where the locations of the study centers, what is the contact information. I would highly suggest that it's used in conjunction with recommendation for with healthcare providers, or at least any results that are yielded through a search be run by your healthcare provider to see if this is suitable. Any of these results yield suitable are suitable for you. So in terms of epilepsy resources, there's epilepsy.com, which would be epilepsy specific clinical trials. And that is not working for me. Sorry about that. Oh my goodness. Oh, it led me to the link. Okay. So can you see this? Maybe not. Okay. There we go. Okay. There is also Cure Epilepsy clinical trials, and again, clinicaltrials.gov. So to just show you what a screenshot might look like, you can see Epilepsy Foundation in the middle here. And what you actually do is you find a clinical trial, you click on it, and actually are able to put in what your location, and so forth. What are you, if you're interested in medication, devices, and so forth. On the left is the Clinicaltrials Gov, similar type of a thing, and Cure Epilepsy on the right. And very similar. So you can say, I live within, okay, I live within 50 miles of Denver, Colorado, and I'm looking for a study on, on seizures, for example. So just a reminder that, of course, this is a great time to be involved. And if you're interested in clinical trials, interestingly, one statistic I found in preparing for this talk is only about 20% of providers discuss clinical trials with their patients, but 70% of patients say they would like to participate in a clinical trial if they were aware that they were available. And so I think this is a kind of a good commentary in terms of being an advocate, a self-advocate, and a patient advocate in November with epilepsy awareness. And so I'm happy to take any questions at this time. Thank you for your attention.
Hey, Dr. McDermott, thanks for the talk. You touched on using diaries as far as tracking seizure counts or other outcomes. I wondered if you could give a little more insight into what factors you're looking for in what makes a good diary, or how that kind of fits into the clinical trial design. Our company has made a few seizure diaries and just wanted to look for opportunities to make them more effective in the future.
So that's a hot topic. So I think seizure diaries are difficult. I think it has to be easy for the patient to use. And when we're talking about clinical trials, obviously there's a recommendation that was specifically in conjunction with the Epilepsy Foundation to have an electronic platform for patients to use. Now, I agree with this. Having been in clinical practice for more than 10 years, I know that if it's overly complicated and requires, passwords and logins and frequent logins, that the ability to do this is going to be, and the kind of yield is going to be low. So I think that electronic diaries are great, but I think that I think they have to be very patient friendly, and have to be very clear in terms of what seizures are being tracked. So, for example, patients have, may have more than one seizure type. And so it has to be very clear to the patient, if there's a coding system, for example. Seizure A is, a drop seizure. And all that the patient or caregiver has to do is like click a button, and it's that easy, right? Or seizure B is a tonic-clonic seizure, and they click a button and do that. I think there should be some sort of surveillance where either the monitor or the sponsor is able to, see whether or not there's a daily entry, if you want good clinical data, and have some sort of like reminder for the patient or caregiver to fill it out on a daily basis, especially in the short term. If there's a years long study, I think that's not feasible, but a 12, 16, 18-week study, I think that should be doable, but it has to have some flexibility for the patient, and has to be reliable.
Got it. Great. Yeah, that makes sense. That's what we've heard as well. And it's one thing to make a tool for doctors to use, and another for the patients to interact with, and being able to be able to have specificity in the data that we're capturing, but also make it very easy to use and simple for a parent to check in, or somebody with some kind of cognitive impairment, right, is a huge part of the design that we're looking for. So I appreciate your input there.
Yeah, definitely. Anybody else? Not at this time. Okay, well, I hope you all have a wonderful afternoon, and thank you for your time.